Hello, everybody, and welcome, and thank you for joining today's webinar at the New York Botanical Garden. I'm Alex McAlvey, Assistant Curator in the Institute of Economic Botany at the Garden, and it's my pleasure to welcome you for today's presentation, Crop Wild Relatives and the Role of Herbaria in Future Food Crop Security, presented by Dr. Makenzie Mabry. At any time during today's event, you're welcome to submit questions by clicking the Q&A button at the bottom of your window, and we will gather these for our speaker to answer at the end of the presentation. Live captions can be enabled by clicking the closed captioning button and then show subtitles or view full transcript. Today's presentation is being recorded and will be available in the NYBG lecture library. So today I'm happy to present Dr. Makenzie Mabry, who's an iDigBio postdoctoral researcher with the Florida Museum of Natural History at the University of Florida. Her research aims to understand the evolutionary processes that produce diverse phenotypes in plants. She received her Bachelor's of Science and Master's of Science from San Diego State University, where she researched the evolutionary origins and history of the plant genus Cryptantha, and earned her Ph.D. from the University of Missouri, where she studied the origins of brassica oleracea crops like broccoli, kale, kohlrabi, and cauliflower. Dr. Mabry is currently working on ways digitized data can be used to inform future crop breeding efforts, especially in the face of climate change. Today, she'll be sharing some of this fascinating work on crop wild relatives and the role of herbaria in the future of food security. Please join me in welcoming Dr. Makenzie Mabry.
Hi, everybody. Can you hear me okay? Hopefully. Alex, can you hear me okay?
Yes.
Perfect. Awesome. Okay, well, thank you so much for that great introduction. And thank you everyone for being here today. I'm really excited to talk about some of my favorite stories and talk about crop wild relatives and also about the role of herbaria. I think there's a lot of great opportunities. So today I'm going to tell you 4 different stories, kind of 4 vignettes, starting with the evolutionary history of brassica oleracea. Then we'll talk a little bit more about crop wild relatives, followed by some feral opportunities, and then we'll kind of shift gears a little bit and talk about persistent identifiers in monography. So hopefully at least one of these stories today will be something of interest to everybody in the audience. And so I wanted to pause here on this first photo or illustration that I'm using for the kind of cover spot for the evolutionary history of brassica oleracea. And so this is one of my favorite products from my dissertation. And I actually did my dissertation with Dr. Chris Pierce, who many of you I'm sure are familiar with when he was still at the University of Missouri. And this beautiful illustration was done by Andy Kerr. She's a fantastic illustrator, a fantastic scientist, and a fantastic educator. And so when we talked to her, we said, you know, we have this funny idea of Darwin at a farmer's market, really enjoying all of the beautiful brassica crops. And this was exactly what she turned to us in the first revision. So it's pretty fun to imagine him walking in and really being impressed with the selection. So it's a little funny pun there. And this was enjoyed by so many that it was excitedly chosen as the cover art when the story was published with MBE. So brassica oleracea, we fondly refer to it as the dog of the plant world, because like dogs, where you have a common ancestor, in this case the wolf, that has led to a lot of different breeds that we know and love today, all this diversity from Chihuahuas to Great Danes. Brassica oleracea has what we think is one common relative that has been selected for all these different vegetable types that we know and love today, including one that hopefully many of you enjoyed last night with your St. Patrick's Day meal. However, unlike the story of the dog, we really don't know who this wild relative is. However, there has been several different species put forth in the literature as what this plant species may be. These hypotheses include from England. So there's a lot of wild cabbage found off the coast of England. So that's one hypothesis that's been put forth in the literature. Another one is that it could be a domestication origin in Italy or Sicily, where the center of diversity occurs for these wild relatives. And then finally, a third hypothesis is most recently from the Eastern Mediterranean, and this is supported by linguistics and literary analysis, as well as a little bit of DNA. And this really focuses on two possible species, Brassica cretica, here found in this yellow area, and then Brassica hilarionis, which is really only found from the island of Cyprus. And this led us to ask two major questions. So one, which wild relative is most closely related to Brassica oleracea? And then where is that center of domestication? To answer this question, we did our best to sample from the many diverse cultivar types. So while you really might be familiar with just 6 different cultivar types, so our broccoli and Brussels sprouts and cabbage and cauliflower that you find in the grocery store, there's upwards of 20 different cultivar types in this one species. So there's all these different beautiful kale types, from curly kale to dino kale, lacinato kale. There's marrow cabbage, which has really, really thick stalks and is fed to livestock. And then there's my personal favorite, which are walking stick kale, which can be over 6 feet tall and are typically then harvested and kind of whittled down to really strong walking sticks. We also did our best to include as many wild relatives from that list of potential wild relatives as we could. And so this is an illustration for some of these. And these are also, I'll use these throughout this part of the talk, and these are all illustrations also done by Andy Kerr. They do look like photographs, but they're illustrations. She's just that talented. And to tackle these questions, we also use a variety of methods. So we borrowed methods from archaeology, we use DNA, we use written sources, as well as some niche modeling. And I wanted to importantly highlight that this took a team effort, a group of international interdisciplinary researchers from across different fields and across the world. So I kind of have split it up into 2 different groups here, although really researchers kind of helped in all the different aspects of this paper. I want to highlight that I got to work with this awesome undergraduate, Evan Gallagher, who's now working on his PhD at the University of Ohio, Miami. Dr. Alex McAlvey was also a part of this project. And of course, like I mentioned, Dr. Pires was my PhD advisor. So he had thought about this question for many, many years. So it's excited that we were able to answer it. So the first result that we were able to digest was that using DNA. So here, what you're looking at is this large phylogeny at the very top. And this is all of our samples in our study and how they're related to one another. And I know it's probably too small to read, and that's okay. I'll blow up the important parts in a second. But what I really want you to focus on is these colored bars right below. And these colored bars are % shared genetic ancestry. So in this case, we asked, what does this shared ancestry look like if we just split everything into 2 groups? So this teal, this blue color is our cultivar ancestry, and this purple is our wild ancestry. So what we can see is that for the most part, our wild relatives that we included in the samples come out as sister to all of our cultivars. So here in that teal, that blue color. However, there's a couple of interesting examples. We then can ask, what does this look like if it's 3 groups? And so then we have this kind of fluorescent green color. And in this group, we find Chinese white kale, broccoli, cauliflower, as well as Romanesco. And then finally, we can ask what it looks like if there's 4 groups. And now we get a unique group of Chinese white kale. And then I've added all the other cultivar types and where they fall out in the phylogeny and what their colored bars look like. But we can dive into some of these interesting patterns as well. One thing is where our wild relatives fall out. So here I'm highlighting just 3 wild relatives, and these are ones that we're keeping on our list as possible progenitor or sister species. These other ones that have been kind of lightened out are ones that are sister down farther in the tree and have only wild ancestry, while these 3 here, Brassica hilarionis, Brassica incana, Brassica cretica, seem to have shared ancestry with the cultivars and the wild relatives. We also find some interesting patterns where some of these wild relatives that we thought were sister species came out as sharing only cultivar ancestry, so meaning that they're feral. And like I hinted at the beginning of the talk, we'll get back to these in a little bit. We can again look more closely at some of these different patterns. So here I'm blowing up where this black rectangle is. And this is 3 species, 2 samples of Brassica incana, 1 of Brassica hilarionis, and 1 of cretica. And what we see that these are the bars at those different grouping levels is that Brassica incana shares 100% shared ancestry with cultivars, no matter how we group them. Brassica hilarionis had about 30% shared ancestry with cultivars, plus ancestry from all the groups. And then Brassica cretica had 60% shared ancestry with cultivars, plus ancestry from all the groups. So we're kind of keeping those 3 species. We've narrowed it down from the long list of potential species, and we're narrowing it down to those 3 now. And then we can also look at some of these other interesting patterns. So here I've blown up again this section in the black rectangle, and this is again 3 species. This is 3 samples of Brassica incana, 1 of Brassica villosa, and 1 of Brassica cretica. And we're calling this wild C. clade 2. And this is an interesting group that seems to be grouping together because it has shared ancestry with cultivars and a little bit of wild taxa. So here that's that pattern of the purple and the blue. So I'm going to keep that group in mind. When we look at those species phylogeny, so kind of a broad overview of how these are related, we see some familiar patterns and are able to resolve some of these questions a little bit more clear. So first we find that cabbage and collards, which are long thought to be sister to one another, are in fact sister, followed by Brussels sprouts. So this is an interesting and closely related group, also delicious. And then we find marrow cabbage and curly kale as closely related to one another. And then we find lacinato or dino kale as closely related to Brassica incana. So this is a potential wild relative that we're finding falling out completely with cultivars. So perhaps indicating that at least our samples of this species are also feral. We then find some interesting groups, and unsurprising cauliflower and broccoli as sister to another, followed by Romanesco. And then we find Chinese white kale, sister to all other cultivars. And this is interesting because Chinese white kale is really the only Asian crop type, while the rest are more European. We also are able to resolve Brassica cretica as the closest living wild relative. So that in mind, we can then further perturb these patterns to look at if we can explain even more of the variance in our data set. We can do that by looking at patterns of admixture. So this is the same, a little bit of a modified, but basically the same phylogeny, just as a different order, different presentation that we just looked at. If we don't add migration, we can explain about 87% of the variance in the data set. However, if we do add migration or gene flow or gene exchange, we are able to explain much more of the variance in the data set, about 92%. And we can look at these a little bit more closely, these events and what they are. So here we're looking at a single event from kohlrabi to a single sample of Brassica cretica, 199, which was coming out with the cultivars rather than where the other Brassica cretica samples were coming out that was closer to the wild relatives. And this made a lot of sense to us right away, because when we grew this accession, this Brassica cretica 199, we noticed that it had a swollen stem. So we can easily see that it's getting that swollen stem from that gene flow from the kohlrabi, which also has a swollen stem. Another interesting was this event from Brassica rupestris to wild C. clade 2. So perhaps this explains what we saw in our shared ancestry or shared genetic ancestry, where these species have this shared integration event from Brassica rupestris. So that possibly it does explain what we see for those. Why it is 3 different species is still something that remains unknown and needs further investigation. We then turn to archaeological evidence to try to further understand this story of domestication. And what we were able to find is the oldest record of that we could find was from seeds, just 3 seeds that were found on a hilltop settlement in the Southern Alps. And these were from the Middle Bronze Age. However, what was important to note here is that it's really hard to tell brassica seeds apart from one another. So here in this image is a penny with a bunch of little brassica seeds. So they're basically just tiny spherical seeds that are usually for Brassica oleracea, pretty much brown or black in coloration. So many folks actually think that these earliest seeds that were found from this hilltop settlement are actually Brassica cretica rather than Brassica oleracea. So then if we try to look at something other than seeds, we can look at residue of brassica leaves, and there are found some that have been identified within pottery shards. And this is from the much later, so in the early medieval area and from England. So we don't find too much additional evidence here for archaeology, and mostly geared because it's so hard to tell the seeds apart from one another. So then we can turn to classical and medieval written sources to try to get a better understanding. So we were able to find much older references. So the earliest textual references came from 2500 to 2000 BP, and this is from Hipponox in Greece, where he refers to a 7-leaf bank cabbage in iambic verse. And this seems to be in referring to either an oath or an offering. So that's a very interesting one. And then following that, just a little bit later, also from Greece, Thorasaurus describes 3 varieties, as well as describes or refers to a wild type that had a bitter taste, many branches, and smaller leaves. And that description sounds a lot like Brassica cretica to us. And then much later, Pliny the Elder in Rome, this is the person, not the beer, if anyone is a fan of the beer, lists a large number of varieties. So now we're seeing this diversity, kind of references to this diversity that we also know of today. And so taking this all together, what we can summarize is that what's the modern day distribution of Brassica cretica includes the Southeast Greece and Aegean Islands, but Brassica incana and Brassica oleracea are not known from Greece. So it is possible that these earliest types that are being described in these textual references are all Brassica cretica. And that's interesting because Brassica cretica itself is also very diverse with multiple named subspecies, and it certainly warrants its own study to kind of further investigate and try to tease these stories all apart. So finally, Alex McAlvey worked to look at what these suitable habitats would be during the time with which we think this domestication event occurred. So this is during the late Holocene. And we focused on this Eastern Mediterranean species because of those textual references. And we find that Brassica cretica really had a pretty widespread habitat suitability, while perhaps unsurprising, Brassica hilarionis is really still restricted to that island of Cyprus. So taking this all together, when we asked which wild relative is most closely related to Brassica oleracea, we find that the data, again, when surmised together, seems to point to Brassica cretica as supported as the closest living wild relative. And that would mean that the center of domestication is really in the Eastern Mediterranean region. So that was an exciting story to further push along, and there is still a lot of places to go back to further investigate this, including additional research on Brassica cretica itself. Okay, so hopefully that also made everyone just as interested as I was after that research on crop wild relatives in general. So in this image here is a bunch of Brassica cretica plants just growing happily on cliffs. There's very different habitat than what one would expect when you're thinking about their crop relatives like cabbages. And might be asking, so just what are crop wild relatives? So if we ask Wikipedia, they say that it's a wild plant closely related to a domesticated plant. It may be a wild ancestor of the domesticated or cultivated plant, but it also could be a closely related taxon. And this is the definition that I tend to use. It's more broad, inclusive definition. And here is just some examples. So here on the right, we have some common crops. So we have sunflower, bean, wheat, and corn, and we have some wild relatives on the left. So they might look a little bit similar, but they might have reduced parts or be many more branched, depending on the species. And what's interesting is crop wild relatives have played a role in domestication since the start of domestication. So this is from a recent paper by Albee et al. In 2021. And so what they're illustrating here in figure A is this early starts of domestication. So in this yellow-orange circle, they're referring to wild populations with low levels of domestication genes. And these green circles, they're highlighting wild populations that have no domestication genes or alleles. And then all these different colors of arrows refer to different levels of genetic exchange. When we move forward in time, you can see that some of these populations start to change to populations undergoing domestication. But importantly, you see that these arrows still exist. We still see gene flow from all these populations. We see it from populations undergoing domestication to these populations with low levels of domestication, alleles or genes. And then we also see exchange happening from the wild populations with no domestication syndrome alleles or genes. As we again move forward in time, we see that these populations are now fully domesticated populations. And still, again, we still see these levels of gene flow, but now we're also seeing kind of the higher levels of genetic exchange between these fully domestic populations. And then finally, these large fully domestic populations expand and we get regular genetic exchange via migration and trading of crops. But really importantly to note is that we still see these low levels of genetic exchange with these wild populations that don't have any domestication genes or alleles. So this is to say that crop wild relatives have always been an important part in domestication, and that really domestication should be thought of as a landscape event rather than this like single event that happened and then stopped. So you might be asking, and hopefully I've convinced you that crop wild relatives are interesting, but now I should also hopefully convince you why we should care about crop wild relatives besides that they're just interesting. It's predicted that we will need to feed 10 billion people, and that's going to take about 56% more food. However, to get that amount of food, we can't use more land. We already use about 50% of the land for agriculture, and we actually want to try to save areas, not use more areas for agriculture. Additionally, we want to lower emissions, so we know we need to fight against the effects of climate change. So here, this World Resources Institute is suggesting, and as I agree, that we need innovative technology. And here they suggest, improved feeds. They use examples of plant-based burgers, which people can argue have their own problems, but may be also delicious. And then what I'm really excited about is I think here with resilient crop breeds, that crop wild relatives and natural history collections can be a huge help and assistance. So I do not think I need to convince this argument, but with this audience, but herbaria are an underutilized resource. And what I mean in this specific context is that when we're looking for wild relatives, we typically go to seed banks and germplasms. And this is showing those wild relatives, again, the same ones that we talked about with Brassica oleracea, and the amount of accessions that are currently available through the Germplasm Resource Information Network, GRIN, and this is sponsored by USDA. So I hope you can see these numbers are incredibly low and very few. So for the last project I talked about, we did have to go to international germplasm, but the story is pretty much the same, just very low numbers that are accessible. However, if we look at herbaria, so here is pulling records from iDigBio, GBIF, and Bison, we have a lot more records. This number is 500 up here, and here's all the same species. So the lowest one is maybe like 25, but we have a ton of different records we can take advantage of. And this is not unique to just Brassica oleracea wild relatives. If we look at eggplant, and these are just some examples, we look at soybean, tomato, apple, corn, rice, wheat, there's tons of records. So this is over 1,500 records for one crop wild relative of apple. So no matter the crop type, there's a lot of resources that we can take advantage of in different ways. And one way might be using the physical specimens themselves. There's really great innovative methods where we can use DNA from these different specimens, and that allows us to answer questions like domestication origins and identification of crop wild relatives. For many crop species, we still don't know who the closest relatives are or where they're originated, so there's a lot of work in that space. There's also really excellent work being done in the lab where I'm currently working, where they used herbarium specimens to study the evolutionary origins of key agronomic traits like nitrogen fixation. They've also been used and can continue to be used for tracking crop pathogens, also studying adaptation, including local adaptation. And then what I'm really interested in is how we can use the digital specimens. And some ways that they've already been used is by performing gap analyses in places that we need to focus collection in. Also can perform niche suitability modeling, predict climate change response, and we can use methods like emerging methods in artificial intelligence, like image recognition and text parsing. And so, like Alex said, what I'm working on for my postdoctoral research and what I'm excited to talk about today is how we can use this digitalized specimen data specifically. And so I work with iDigBio, so this is the integrated digitalized biocollections. And what this is really a national coordinating center for digitalizing biodiversity collections. It mainly serves to ingest, integrate and serve data, and we can pull data from localities, we can pull dates, we can pull images, all sorts of great things. And here is an example that's more specific to plants. So iDigBio, in that resource, you can find fossils, you can find insects and birds and mammals. It's a natural history collections at large. Looking more specifically at herbarium specimens, one thing that we can pull is just this information from the label. So here in the red circle, we can pull things like the scientific name of the plant. We can pull the date it was collected, who collected it, where it was collected from, any other associated species, as well as any notes that might have been included. And so I'm going to show a quick model of how we can do and perform ecological niche modeling of crop wild relatives of Brassica oleracea, and I'll focus this on one example with Brassica cretica. So if we pull records for Brassica cretica from their repositories, so here's iDigBio, we're able to pull 11 records. While that seems like a little bit, it's still a lot more than in the germplasm. And in the GBIF, we're able to pull 500 records. So total raw records we're able to pull of digitalized collections is over 500. However, after we clean it and removing duplicates and any locality that don't have any records that don't have locality, we get about 150 records. And while it seems kind of disappointing going from 500 to 150, I still am really excited because this is just so much more data to work with. And so what we're able to do is make these niche models or habitat suitability models for each of these species. And this is similar to the ones I showed you for Brassica cretica earlier. However, this is its current model. What we showed in the first part of this talk with looking at how we use niche models, we're projecting back in time. So when we thought the domestication event occurred, and this is different right here, this one right now, because this is where we see it today. So the red dots are the localities that I used. And so perhaps unsurprisingly, it's darker blue where more of those dots are. So this species really likes the islands. And then as you get to farther away from these localities, they're less suitable habitat. And I can't forget to acknowledge that this work would not be possible without this awesome undergraduate that I'm working with, Tori Ford, who is just a whiz in figuring out computational analyses, also was responsible for making this beautiful map. So an excellent undergraduate researcher, and I feel super fortunate to be working with them. What we can do with these maps, and Tori is helping with this, is then project them into a global space. And this allows us to test for global niche suitability. So perhaps there's other environments across this global world that Brassica cretica would also do well in. And so that is important information if we're thinking about which crop wild relative may be best for different genome editing techniques. But what we also can do is then take those models and project them to various climate scenarios. So here is what is a few different models that have been developed. We would be here, about here right now, and then depending on how we act as a society using different shared socioeconomic pathways that have been developed will depend on what our greenhouse CO2 levels may be in the future. So for example, if we go with a middle of the road where we kind of have middle support for adaptation and middle support for mitigation. So we use this SSP2 line. We would probably follow something along this gray line. At least that's what's modeled. So it might stay about the same and eventually may get better. However, if we really don't do much and continue to use fossil fuels, we might end up being somewhere more along the red line. And so we can take all these models and project our habitat suitability to get an idea if there's a crop wild relative that might have more niche suitability than others when we take into consideration these climate scenarios. And this is, again, exciting because with neo-domestication, we can take advantage of genome editing techniques, and that allows us to use crop wild relatives which best suit our needs. So maybe one crop wild relative that's not sister may do better in spaces where we're having trouble growing crops. So we can use that wild relative to introduce that variation, identify that variation using genetic techniques, and then hopefully introduce it to make better crops that will perform better in that space. Neo-domestication is much quicker than conventional breeding. We also no longer need to worry about crossability. So this is an exciting time and makes it perfect for taking advantage of these resources in the herbaria. However, what about other sources of diversity? So hopefully this figure looks familiar. It is the same one from talking about the evolutionary history of Brassica oleracea. And I've kept two species highlighted, so Brassica encana and Brassica oleracea. And what we found from those earlier analyses are that these were feral crops. And I think that these are an exciting idea and source of diversity when we're thinking about future crop improvement. And so that is what we will talk about next. So this is a picture of some feral Brassica oleracea on an island. So it really does like that cliffs, but I think it has a pretty good view as well. So what is a feral crop? So one of the most recent definitions for a feral crop is it's a de-domestication or feralization, which is an evolutionary process by which domesticated crops or livestock reacquire components of wildlife traits and escape from intensive management by humans to form independent reproducing populations. And so what that might look like, for example, is here we have a cabbage plot or a cabbage patch, and it may escape from that cultivated space and persist and eventually acquire these wild-like traits and become what we call endoferal. Another pathway could be that a cabbage escapes and then crosses with another wild plant that's living nearby and become what we call exoferal. So multiple pathways to becoming feral, and exciting to think about the different ways we could take advantage of those different types of ferality to think about crop improvement. We surprisingly know very little about feralization, and some of that comes from some negative stigma associated with it. But we really have no idea of how many crops have feral populations. We know that there are over 250,000 plant species that are known, and about 30,000 of them are edible. However, only 7,000 have been used for food, and only 120 are cultivated today. And of those 120, really only 9 provide about 75% of the human diet. So this is a very small percentage of what's available already. And then within this already very small percentage, we really know a little bit about them. So how many feral populations? We do know of some. So here is a quick word cloud of some of the species that have known feral populations. And these span different types of grasses. These span different types of trees. There's vegetable types, there's fruits. It really doesn't seem to be, at least yet, any specific patterns that we can tell. That's a space that we're excited to hopefully continue to investigate. But thoughts on feral have been around for a while. Even Darwin in 1868 had a quick jaunt about ferals in his Variation of Animals and Plants Under Domestication. He writes, in our turnip and carrot beds, a few plants often break. That is, they flower too soon and their roots are generally found to be hard and stringy, as in the parent species or that crop wild relative. By the aid of a little selection carried on during just a few generations, most of our cultivated plants could probably be brought back without any great change in their conditions of life to a wild or nearly wild condition. So he's talking about fertilization here, although he doesn't say fertilization straight out. So it's interesting that this concept has been around and has been acknowledged, but we still know very little about it. So together with Alex, we last summer organized a colloquium where we worked with this awesome group of individuals as well as attendees of the colloquium to further kind of discuss what we know about fertilization and crop wild relatives. And we came up with several large topics of open questions in fertilization. One of the major ones is timing and frequency. So how frequent do feral crops occur? Is there anything that's driving this? And this is one example of feral populations that are known in just Brassica-oleracea. So we can find them off the coast of Chile, off the coast of California, all across the UK, France, Portugal, Spain. And this dot in the middle here is actually where that cover image is taken. So this island near Antarctica. So very interesting to think about how it got there and how frequently this is occurring if this has happened all across the world. We're also interested in trying to dissect the genetic architecture and pathway of fertilization. So this is from a recent review that I wrote with actually Troy Rowan, who is a cattle genomicist. And so just like plants have fertilization, we know animals have fertilization. So beyond just understanding this plants, there's even bigger questions about understanding this in organisms broadly. But we're interested in is, does fertilization happen the same way every time? Are there patterns that are consistent? So here we have a quick illustration of hybrid fertilization and different population alleles, as well as different endopheral events. And then the location, can we see if location matters? Can we find any signals of local adaptation? We're also interested in further dissecting the evolutionary history. So this figure back to Albee et al. In 2021 talks about these populations, which they term wild populations with low levels of domestication syndrome alleles. This sounds a lot to me like a feral population. So can we tease apart how ferals historically have always played an important role in domestication? Also interested in how these feral populations and if these feral populations can be used in agronomy and breeding. Interested in the effects of feral populations. A lot of them have negative stigmas as being invasive. So what are the effects on ecology? Additionally, the negative stigmas against these populations have occurred because of conservation efforts. So this is from a study by Green et al. In 2015, where they identified these pink dots that were feral populations that had trans genes in them in seed and hay alfalfa production areas. So there's conservation concerns and how can we mitigate that? And then finally, we're interested in seeing, can we talk about and describe best practices and methods so that we're working together as a group to digest these big questions in fertilization. So rather than having turnip fertilization specialists or rice fertilization specialists, can we work together to really get at these core pathways and functions? And when we go looking for feral crops, the story is very similar to crop wild relatives. So here I've added a row to this table from the Germplasm Resource Information Network. And so currently available, there's 3, what they're saying are wild Brassica-oleracea or feral Brassica-oleracea populations available. So just 3 samples. And of course, we can look for them in the herbaria. And so I was incredibly surprised to find that when I look for crops in a herbaria, again, from looking at repositories that include iDigBio, GBIF, and bison, there are thousands of collections. So here's for Brassica oleracea, and we have about 1,000 collections, even corn, wheat, eggplant, tomato. This is just a few examples, and we can do this for many different crops. There's a lot of samples in herbaria, which were really surprising to me because I can't imagine too many botanists out collecting in the middle of like cultivated fields. So what this opens up is an opportunity to explore these collections to see if these are found outside of cultivated zones using the information that's within these records. We can then do very similar things that we did with the crop wild relatives. We can use the physical specimens. We can pull DNA and look for feral origins and identification. We can also include them for studies of adaptation and local adaptation. We can then additionally, just like with the crop wild relatives, use the digital specimens for looking at niche suitability modeling. Do these feral populations provide any more information than the crop wild relatives? Can we compare it to what we know about the crops that are growing right next to them? We can also then see if they perhaps are predicted to do better in climate change now that they're outside of cultivation and have adapted to that space. And then we can also use emerging technology of the artificial intelligence for image recognition. So there's really great work in trying to understand phenology changes, and we can do the same thing with these feral populations as well. So this last little bit, I did promise I would kind of turn, but not completely, because I want to talk about how monographs can act as a nexus for building extended specimen networks using persistent identifiers. And why this relates is that we're talking about broadly for crop wild relatives using these digitalized collections from herbaria is natural history collection enabled research. And to be able to do this efficiently and effectively, we must improve the findability, the accessibility, the interoperability, and the reuse of digital assets. And this follows the FAIR guiding principles for scientific data management and stewardship. So this fits into those stories. To do this work with digitalized data, we want to be able to find it easily and take advantage of it to the best of its ability. So for those of you that might be less familiar, monographs are where we can compile, integrate, and interpret the information associated with specimens that are stored in biological collections like herbaria. So here is an example of a monograph of this section, Eukarya in Cryptantha by Payson from the Annals of the Missouri Botanical Garden. And this can include a variety of different types of information. It can include phenotypic data, it can include genetic data, geographic data, ecological data, and even phylogenetic trees. And together, these data are important as taxonomists use them. It's representing taxonomist opinions and decisions that's made by systematists. So these are important documents that are really foundational. Now, on kind of the same hand, but a little bit different, we also have the extended specimen network. And this is, I'm sure, not unfamiliar to most of you as well. And this is a great example of what's being proposed. So here we have, again, our physical specimens with digital images and records. And these are a primary extension from that physical specimen that we might find in a herbaria or any biodiversity collection. And then you have additional extensions from those specimens. So you might have molecular data or morphological data. And then you also might have like species distributions or different niche models that are formed from those specimens. And so these can all be connected. We get a lot of information when they're connected than when they're kind of individually separate from one another. And this led this awesome group I got to work with through a workshop where we asked how we can modernize monography to suggest monographs as a nexus for building extended specimen networks. You hopefully saw that these two resources, the monograph and these extended specimen networks, have similar data overlap. So can we use these in new ways? So this is a great group of co-authors that I got to work with that came from a variety of backgrounds that made this a really fun project. And what we suggest is that since taxonomic monographs already act as kind of this hub where specimen data are stored, that when we think of future monographs, we should try to incorporate them or use them as portals for this resources of linked biodiversity data that's embodied by the extended specimen network. However, key to realizing this is we need to shift from kind of this traditional taxon-based monography to specimen-based monography. And then finally, we need to adopt citing both known identifiers and persistent identifiers for specimen and all metadata and data that's associated with those specimens. And this is just a artist rendering of what an artist we worked with that kind of to visualize what we're thinking. So again, these are specimen-based. So now we have the specimen at the center of these monographs rather than the taxonomic group and having specimens kind of at the end as a list. They're front and center. And we might have fossil specimens, we might have physical specimens, and then we have all the associated data, whether there's a digital specimen record or media file. You also might have genetic data that has gone into making another type of associated data like the phylogeny. So hopefully this is sounding very reminiscent of that extended specimen network. You also have identifiers, ecological data, and each one of these circles has these little identifier tags here illustrated using QRS codes, but they're unique identifiers. They don't have to be QRS codes, but these are just to kind of represent that. So what are these persistent identifiers? A persistent identifier is a long-lasting and globally unique reference to a digital resource, and it can't be used to refer to a different object. Sometimes these are treated as synonyms of globally unique identifiers or universally unique identifiers, but the really important part is the persistent. And this is really challenging for both social and technical reasons. So an example of what this might be that folks are maybe more familiar with is an ORC-ID. So this is a unique ID, a persistent identifier associated with different researchers. And this is different than using known identifiers, something like a specimen number where we have CAS 1, 2, 3, 4, 5. And this is not unique because although it may be persistent, but it may not be unique because this could refer to the California Academy of Sciences, the Chicago Academy of Sciences, the China Academy of Sciences, and it could be a collection in the herpetology department, it could be a collection in the mammalogy department, or it could be a plant. We really don't know. So that's where the difference comes. And this group of folks were perfect for trying to tackle why this hasn't been adopted, because we're not the first to think of these ideas. Like I showed you that extended specimen network has been an idea in the literature for a while, but why has it not been adopted by researchers? And so with this group of folks, we have backgrounds that are practicing systemicists, that are not excited to use taxonomy. We're a group that are biodiversity informaticists, specialists that aren't practicing taxonomists, and all different types of organisms from mammalogy to fossils to plants. So that really gave us an ability to dive into the question is why this has not been adopted yet. And what we kind of the overall thing we identified is that there's this parallel universe where we're practicing. We have taxonomists on one side and then biodiversity informatics specialists on the other. And we aren't seeming to be talking the same language. So instead of trying to get to that perfect, monograph enabled or persistent identified enabled monograph, let's take a first step as a community. And so what we suggest is adding a Darwin Core archive file with monography, just as a first step. And so what this would look like is we have our specimens mapped to Darwin Core codes. So if folks aren't familiar with Darwin codes, they have a format that looks, these are different examples here. And so we have a Darwin Core and scientific name, followed by the different specimens. And I hope you can see already that there's a bunch of different formats of how this data has been put in these repositories. So here, this catalog number includes the institution code. Here, the collection code is an institution. Here, it's a collection. How the authors are put in is different. Here is the ORC-IDs that are used to identify who identified the plant. And then here's some unique IDs as well. This occurrence ID is also helpful, but then also you'll see different formats. So this is one small step forward we think we can make as a community, and just including this as a supplementary file with monographs enables them immediately to be machine readable. We follow this up with additional action items for different user groups, including how researchers can, along with publishing these Darwin Core Archive files, develop biodiversity informatics tools together with different groups of researchers. We hope that museum and collection staffs can be supporting and encouraging researchers to use collections and sending guidance on how they expect the specimens to be cited. Just as specimens need curation, these persistent identifiers will also need active curation, which of course is going to require money and support. And then including and integrating this information into undergraduate and graduate coursework will be an important part to get it accepted. We hope that repositories can serve as help desks for existence, as well as developing bioinformatic tools and implementation of the persistent identifiers. We hope publishers can help to make persistent identifiers required for publishing and then add tools to the publishing process to support this and not making it harder, but hopefully to support it. And then we hope in the future that funding agencies can create a central registry for minting, storing, and linking these identifiers. And these are all kind of what we hope for the future, a dream scenario. And then finally, we hope that professional organizations and meetings will support training events and sessions on these topics. And there's a broad amount of places that these could occur. So hopefully today I've been very grateful to share some of my favorite stories with you all. But the first one, just to remind everyone, we talked about was the evolutionary history of Brassica oleracea, where we found that Brassica cretica was supported as the closest living wild relative, and that it likely had an origin of domestication in the Eastern Mediterranean region. I hope I've convinced you that Herbaria have many collections of crop wild relatives that we can use to inform future food security, that feral crops provide important pools of diversity for future crop improvement, and that using persistent identifiers in monography can enable the discoverability and reusability of biodiversity data in novel ways. I, of course, want to acknowledge all the listed co-authors throughout this talk, which, of course, without them, none of this work could be possible. I think it's always important that science is a team event. It's great to work with others. And of course, I also want to thank those that supported this work, both at the University of Missouri and funding from the USDA, and current researchers at the University of Florida and funding from NSF and iDigBio. And with that, I'll be happy to take any questions, and I thank you for everyone's attendance.
All right. Thank you very much, Makenzie. That was very thought-provoking, and it's great to see the directions your work has been taking. I think we have some great questions here from the audience that we can cover in the next few minutes. So one question is hybridization known or suspected in any of the earliest crop development?
I think that's a good question. I think there is a lot more recent evidence that for many crops that we're seeing, we're able to kind of tease apart some of those patterns. But I think for many crops, that signal of integration, even if it occurred, probably will not be findable in their current genomes. But I think it's probably more feasible than to think that things were separated and completely in isolation when we think about human history alongside of it.
And you've done some work with these polyploid crops too, which is kind of a different style of hybridization. Maybe you can speak a little bit to that as well. The role of allopolyploidy in crop domestication.
So if I understand correctly, kind of asking about, many of our crops, our current crops have a polyploid origin. Is that what you're referring to?
Yeah.
So that is like additional evidence that it's likely that multiple, if not most, many of our crops have an integration history.
Great. So here's another one from Paulo Gaero. how do you predict that fertilization could affect diversity in wild crop relatives populations? In other words, that they could cross and introgress genes from domesticated plants, or that feral populations can even replace them because they are more competitive.
So, okay, I see the question, so I'm just reading it. How can you predict the relationship? So I think there are some examples of this where we do see, like we do know the history of some feral plants. So especially in rice, there's been like the most work in fertilization, I would say. And they know for the different populations of feral rice, they have different histories. So some where they cross with domesticated plants. And then in those systems, there are some examples where it is bad and they do replace the populations, the cultivated populations. So I'd say that it definitely, I think that's why this is exciting, is that we have that one example, but is that the story for all the crops? And so that's where I think really a lot of the work that Alex and I are excited about is like, can we get more examples of this? Is it always bad? If it's always bad, then we need to think about that. But we think there might be an opportunity to use this for our advantage.
Great, yes. Here's another question from Yash Pal Sharma. Excellent talk about the urban environment influence / adaptation of Plantago species. Is this a step toward fertilization? Oh, it's referring to a recent publication, it looks like, this week's Science Journal reports.
So if I understand correctly, so it looks like there was a report, I haven't seen it yet, where Plantago has adapted to more of an urban environment. And I'm not specific on where they came from. If they escaped from a field or like a cultivated situation, then definitely. And is also another really interesting avenue is like if these feral escapees do escape and do better in a urban environment, even more of a reason to think that maybe these are a positive space that we should focus time and energy and money on when we know about what's going on in the world. Yeah, I'm gonna have to look that up. Thank you.
Great. Here's a nice one down here. Could you please elaborate on the difference between endo and exoferal?
Yeah, definitely. There is. So I kept it simple. In fact, there's exoferal and there's endoferal. There's like every combination that you could think of. But basically, what it boils down to is how things became feral. So was it just a single, plant or a few plants that escaped, or somebody left a field, they abandoned a field and it was no longer under cultivation, and those established and did well, we would consider that an endoferal. So from that same crop population. An exoferal is that outside crop wild relative contributing to that process.
Great. And as you said, that might have consequences for how it might be used for breeding or something.
Yeah, yeah. And we don't know for most, so there's all those different species that we know have feral relatives. For many of them, we don't know what their pathway to fertilization was. If they're endo, if they're exo, we really don't have that information to make any true large statements yet. But they're exciting. Yeah. It's lots of space. If anyone's interested, we're always looking for friends.
It's amazing how many unknowns there are compared to the crop domestication or crop side of things. So it's a very wide open field, I believe. And everyone's trying to catch up with the rice people who are way ahead in understanding evolutionary facets of virality. All right, do we have time for more questions? Or I think maybe let's take one more quick question. Thank you so much for your talk. I'm an artist and gardener and have a few questions. First, is the iDigBio herbarium collection open source and available to the public?
Yeah, so if you search iDigBio, you just write that in the land, that portal will come up and you can look through the entire portal. You can also interact with it through coding, but it's a searchable portal as well. It's really fun to search through things in there. Some really nice images as well. So if you're looking for things to base some art around, it's a good place as well.
Yeah, and you saw illustrated beautifully the intersection of art and brassica already with Andy Kerr's work that Makenzie
So
shared.
beautiful. Mm-hmm.
All right, well, thank you everybody so much, and thank you very much, Makenzie. This was really a lucid and enlightening talk, and look forward to seeing more of your work. I think you hit on a lot of the interests of our audience and people interested in herbaria and monography and food and domestication. And so I really appreciate you joining us, and everybody have a nice day. If you're in New York, it's a very beautiful day out, so hope you get outside.
Thank you for being here, everybody. Thanks for this opportunity.